Welcome back to the deep dive. Today we're doing something a little different. We aren't just skimming the surface of a medical paper. We are practically reading the cellular blueprints of gastric cancer line by line.
Yeah, we really are. It feels less like a standard clinical trial update and more like a microscopic espionage thriller, to be honest.
It totally does, because we're decoding this massive study published in Cancer Cell by Zhao and colleagues, and it focuses on the NeoSummit-01 trial. The central mystery we're tackling today is this: we have this super weapon treatment called neoadjuvant immunochemotherapy.
Right, NICT for short.
Exactly, NICT. And it works absolute miracles for some patients, but then it fails completely for others. I mean, for more than 50% of patients, the tumor just shrugs it off.
Which is incredibly frustrating in the clinic. So the mission of our deep dive today is to open up the black box of the tumor microenvironment to understand why.
Why it works, why it fails. And unlike older studies that just, took a snapshot of the tumor before and after surgery, this team took a total surveillance approach.
That's a great way to put it. They didn't just take pictures, they essentially filmed a movie. They use multi-omics, meaning they sequenced the DNA, the RNA, the proteins, and they even did single-cell sequencing on paired samples.
So they are watching the tumor evolve and fight back in real time during the treatment.
Exactly. It is a level of resolution we haven't really had before.
Okay, let's unpack this systematically. Before we get to the whole spycraft aspect, we need to establish the baseline for you listening. What exactly is this super weapon NICT, and how does it compare to the old way of doing things?
Right. So the old standard is NCT, which stands for neoadjuvant chemotherapy. That is basically just giving the patient systemic chemo drugs before surgery to try and shrink the tumor down.
Standard protocol?
Standard protocol. But in this specific trial, the pathological complete response rate, which basically means did the tumor entirely disappear under the microscope, was only about 20.4% for chemo alone.
So basically 1 in 5 patients gets a great result. That's pretty grim.
It is grim. But in the NICT group, they added a PD-1 inhibitor called toripalimab to that chemo backbone. And this combination pushed the response rate up to 44.4%.
Wow. Okay, so we are more than doubling the success rate. That is massive. But the paper mentioned something really fascinating about a genetic cleanup effect. What does that actually mean at the cellular level?
This is where the multi-omics really shines. When they sequenced the DNA of the tumors after the treatment, the NICT group showed a really significant reduction in major cancer-driving mutations.
Like which ones?
Specifically in genes like TP53 and CSMD3.
Wait, TP53, that's the famous guardian of the genome, right? Like when that mutates, cells stop fixing their DNA mistakes and cancer just runs wild.
Exactly. So the fact that the mutation burden of TP53 dropped so dramatically suggests that the immunotherapy wasn't just shrinking the tumor randomly, it was specifically hunting down and wiping out the clones of cells that carried those exact dangerous mutations.
It's like a deep clean for the genome.
Yeah, chemo alone just kind of bombs the area, but the immunotherapy acts more like a targeted strike team cleaning out the worst offenders.
That brings us to this concept of temperature that I saw in the notes. Turning cold tumors hot. I feel like this is a really crucial concept for everyone listening to visualize.
It is. Think of a cold tumor or an immune desert as a fortified castle. The gates are locked tight, the immune system is pacing around outside the moat, but there are absolutely no soldiers inside the walls.
And standard chemo.
Standard chemo is like throwing rocks at the castle walls from the outside. It might chip the stone, it kills some cells, but it doesn't open the gates.
The combo therapy changes that.
Completely. The NICT kicks the doors down, it converts the fortress into a battlefield, it turns the tumor hot, meaning it becomes immune inflamed. The study showed this massive shift where patients moved from that desert state to a highly inflamed state where T cells were just flooding into the area.
Okay, and here is where I actually found a paradox in the data. The study says that NICT upregulated immune checkpoints, specifically things like CD96, TIGA-T, and PDCD1. But isn't PDCD1, which codes for the PD-1 protein, isn't that the brake pedal for the immune system? Why would we want more brakes?
It does sound completely backward at first glance, but you have to look at the context of what's happening. High expression of those checkpoints is actually proof of life.
Proof of life? What do you mean?
It means the T cells are physically there, they are active, and they are actively trying to engage the tumor. The tumor is panicking and trying to slam on the brakes, sure, but the fact that the brakes are so visible means the car is moving, the battle is happening.
Ah, I see. And since we are giving them a PD-1 inhibitor drug as part of the therapy.
We are cutting the brake lines.
Right.
So the army is inside, the tumor frantically tries to stop them by throwing up checkpoints, but our drug overrides it. That is the fundamental reason why NICT works so well for those who respond.
That makes perfect sense. However, despite all of this, doubling the response rate, cleaning up the DNA, heating up the tumor fortress, it still fails half the time. And the standard tests that doctors use right now to predict success, those fail too, right?
Right. Usually, oncologists rely heavily on TMB, or tumor mutation burden. The clinical theory is simple. The more mutated a tumor is, the weirder it looks, and the easier it is for the immune system to spot it and kill it.
But in this trial?
In this trial, TMB failed completely. It couldn't predict who would survive or who would respond to the combo. Neither could PD-L1 status. The old maps were essentially useless here.
So we desperately need a new map. Let's pause right here for our first checkpoint to lock this in for you listening.
Good idea.
If you're taking notes, here's a quick recap of the landscape so far. First, the stats. Chemo alone gets roughly a 20% response. Adding immunotherapy bumps it to over 44%. Second, the effect. The combo acts as a genetic deep cleaner, scrubbing out TP53 mutations and turning cold fortresses into hot battlefields.
And third, the problem. Traditional predictors like TMB do not work for this specific treatment context. We are flying blind.
Exactly, which leads us directly into the hunt for new biomarkers. Since TMB failed, the researchers had to go fishing in their multi-omics data for new genetic clues.
And they found some very specific suspects. Let's talk about the good guy first, which is FDXR.
FDXR?
Yes, FDXR is a gene you really want to see active. High expression of FDXR correlates with much better survival. It's mechanistically linked to p53 function and overall immune activation. If your tumor has high FDXR, your cells are basically waving a giant flag shouting over here to the immune system.
Okay, so high FDXR is a green light. What are the red lights?
There are three main villains they pulled from the genetic data. First is SBS19. This is a specific mutational signature that highly correlates with a poor response. Second is the FGFR pathway. High activity in this signaling pathway basically distracts the immune system and keeps the tumor cold.
Got it. And the third one sounded like a license plate number when I was reading it. HLA-B 15.02.
Yeah, HLA-B 15.02. This is fascinating biology. The HLA system is essentially the hand that holds up the wanted poster of the cancer antigen so the T cells can recognize it.
Okay, I like that analogy.
Right, so patients with this specific 15.02 allele seem to have slippery hands. They simply cannot present the cancer antigen effectively. The immune system patrols right past the tumor without even seeing it because the wanted poster fell down.
That is incredibly specific and a little terrifying. But the study didn't just stop at bulk genetics. They went down to the single cell level to map out what they called ecotypes.
This is really the visual core of the paper. They realized that gastric cancer isn't just one monolithic thing. Based on the single-cell data, the tumor microenvironment forms 5 distinct types of neighborhoods or communities. They labeled them EC1 through EC5.
I want to walk through these neighborhoods because they basically dictate the patient's fate. Let's start with the best neighborhood to be in. EC1.
EC1 is the immune hotspot. If you looked at this under a microscope, it's an absolute party for the immune system. It is packed with CD8 positive T cells and natural killer cells. If you are a patient, you want your tumor to live in EC1. These people respond beautifully to NICT because the army is already deployed on site.
Okay, so EC1 is the war zone where we are winning. Moving down the list, what about EC2 and EC3?
These are kind of the transitional suburbs. EC2 is defined by tertiary lymphoid structures. These are little localized immune outposts being built right next to the tumor. EC3 is the angiogenesis neighborhood, defined by lots of new blood vessels forming. Both of these show a moderate response to treatment.
Makes sense. Now we get to the rough parts of down, EC4.
EC4 is the fibrosis neighborhood. It's characterized by heavy extracellular matrix organization. Think of it as the tumor being walled off by thick, dense scar tissue. It's physically hard for the drugs and the immune cells to penetrate that wall.
And finally, the ultimate antagonist of our story today, EC5.
Right. EC5 is what the study officially calls the metabolism TME, but I like to call it the bunker. In the trial, patients with an EC5 tumor environment were statistically unresponsive to chemo and totally unresponsive to the superweapon immunochemotherapy.
So they are completely, entirely resistant.
Almost entirely. It is a highly, highly immunosuppressive environment. Nothing gets through.
Let's do a quick check-in right here because Understanding these specific neighborhoods is so crucial for the mechanism we're about to explain.
Definitely.
So for you listening, try to visualize the stomach lining divided into these distinct zones. You have EC1, which is the war zone. That's good, high T cells. Drugs work great. You have EC2 and 3, the middle ground, moderate response. You have EC4, the scar tissue wall, which is hard to treat. And then you have EC5, the bunker. Highly metabolic, highly suppressive, nothing works.
And remember, the genetic keys that map to this FDXR is your green light, while the HLA slippery hands and the FGFR pathway are your red lights.
Perfect. So here is the million-dollar question that the researchers spent the bulk of the paper trying to answer. What exactly is happening inside EC5? Why is the bunker so invincible?
This is where the detective story turns into a full-blown spy thriller. The researchers zoomed all the way in on EC5 using their single-cell data, and they found a very specific cell type that was orchestrating all the resistance. They found APOA-positive tumor cells.
Okay, wait. APOA1, apolipoprotein A1. Isn't that related to cholesterol, like the good cholesterol they check in my annual blood test?
It is exactly that. It's the major protein component of HDL cholesterol.
So why on earth is a stomach cancer cell manufacturing cholesterol transport proteins?
That is the disguise. These specific tumor cells are described in the paper as enterocyte-like.
Enterocytes being intestinal cells, right? The ones that absorb nutrients and fats from our food.
Correct. Now, the stomach and the intestine are neighbors, obviously, but they have completely different jobs. These gastric cancer cells have somehow evolved to mimic the biology of healthy intestinal cells. They basically put on a costume. They start heavily expressing genes related to lipid metabolism and cholesterol transport.
So the cancer cell puts on a fake mustache and says to the immune system, hey, nothing to see here. I'm just a harmless intestinal cell processing some dietary fats.
And it works beautifully. But they don't just hide. That wouldn't be enough to create the bunker. They actively bribe the police force.
How do you bribe the immune system?
You target the macrophages. Macrophages are the big eaters of the immune system. Their normal job is to patrol the tissue, eat dead cells, and clean up cancer cells. But these APOA1 positive tumor cells start sending out lipoproteins, literally little packages of fat, directly to the macrophages.
They are feeding them.
They're heavily feeding them. These lipids bind to a very specific receptor on the surface of the macrophage called TREM2.
TREM2. Okay, so the macrophage eats the fat packages via this TREM2 receptor. Then what happens?
It reprograms the macrophage completely. The macrophage becomes metabolically exhausted. It essentially goes into a massive food coma. Instead of attacking the tumor, it transforms into an immunosuppressive state, often called an M2-like state, and starts releasing chemical signals that tell all the surrounding killer T cells to stand down.
That is incredibly devious. The cancer mimics a nutrient-absorbing cell, feeds the immune system a bunch of fat to make it sluggish, and puts the entire defense network to sleep.
It's a localized metabolic conspiracy. And the researchers didn't just guess this from the sequencing data, they proved it mechanistically.
How did they prove it?
They used organoids, which are mini tumors grown in a dish, and they used mouse models. When they genetically forced the tumors to overexpress APO1, the anti-PD-1 therapy immediately stopped working. The drug became totally useless.
Because the macrophages fell asleep, so they found the exact cause. Did they find a fix for it?
It did. In those same mouse models, when they blocked that TREM2 receptor, essentially putting a lock on the macrophage's mouth so it couldn't eat the lipid bribe, the resistance was totally reversed.
Wow.
Yeah, the immune system woke back up, the T cells reactivated, and the tumor shrank.
That is a massive breakthrough. It implies that for those EC5 bunker patients who currently have zero hope, there is an actual specific drug target.
Exactly. It moves us from saying we don't know why you aren't responding to saying we know the exact lipid pathway and we can potentially block it.
Let's recap this mechanism for you listening, because it is the most dense and definitely the most important part of the entire study.
Go for it.
So why does the EC5 neighborhood resist the superweapon? First, the villain, APOA1 positive tumor cells. Second, the disguise. They mimic intestinal cells to process lipids. Third, the bribe. They feed fat to the immune system. Fourth, the accomplice. TREM2 positive macrophages eat that fat via the TREM2 receptor.
And the result is that the macrophages enter a food coma and shut down the entire localized immune response.
Exactly. And the fix. Block TREM2 to stop the bribe and wake up the army.
Perfectly summarized.
Okay, so we have the genetics, we have the neighborhoods, and we have the spy mechanism. But we need to bring this back to the clinic. If I am a doctor, I can't just tell my patient, hey, you're in the bunker, good luck. I need a treatment plan.
Right, and that is the final piece of this paper. The researchers integrated all this data, the DNA, the RNA, the single-cell interactions, into a streamlined clinical model. They call it MOS, which stands for multi-omics stratification.
And this algorithm divides patients into 3 practical categories.
Yes, MOS1, MOS2, and MOS3. This is the ultimate so what for patient care. It's an actionable decision tree.
Let's start at the top with MOS1.
MOS1 corresponds strongly to that EC1 neighborhood we talked about. These are the responders. They have high immune activation. They often have PIK3CA mutations, and they have excellent antigen presentation.
And the clinical strategy for them?
Don't overthink it. Give them the NICT immunochemotherapy. The response rate in the MOS1 group is around 88%. These are your massive success stories.
88% is incredible for gastric cancer. Okay, what about MOS2?
MOS2 is the middle ground. These tumors are generally cold. They usually have wild-type TP53, so no mutation there, but they have a completely different problem. They are resistant to the chemotherapy part of the cocktail.
Why are they resistant to the chemo?
Their cells pump out high levels of drug metabolism proteins, specifically from the AKR family, the aldo-keto reductases. Basically, the tumor cells are just really, really good at detoxing. They wash the chemo drugs right out of their system before the drugs can cause enough DNA damage to kill them.
So giving them standard chemo is essentially just poisoning the patient for absolutely no reason.
Exactly, unless you change the tactic. The researchers suggest that for a MOS2 patient, you need to combine the NICT with AKR inhibitors. You have to purposefully disable the tumor's detox system first so the chemo can actually stick.
That makes total sense. And finally, MOS3. This corresponds to our bunker, the EC5 neighborhood.
It does. MOS3 is the hardest case. They have high APOA1 expression, high FGFR activity, and extreme immune suppression. In the trial, giving these patients the expensive immunotherapy provided absolutely no significant benefit over just giving them basic chemo.
So for these patients, the current superweapon combo is literally a waste of time and money.
As it currently stands, yes. It just exposes them to immune-related side effects with no payoff.
So what is the new strategy for a MOS3 patient?
The model strongly suggests targeted therapy. You can't just try to blindly boost the immune system because the immune system is actively sedated by that lipid bribe. You have to break the suppression mechanism first.
Right.
The study points to targeting NR5A2, which is a nuclear receptor that regulates that APOA1 disguise, or using specific FGFR inhibitors. You essentially have to dismantle the bunker before you can even think about sending in the troops.
This is what precision medicine is actually supposed to look like.
Yeah.
Instead of a one-size-fits-all protocol where we just cross our fingers, it's a completely tailored suit based on the cellular biology.
Precisely.
I want to put you, the listener, right in the oncologist chair for a second. A patient walks into your clinic. Based on this study, how do you treat them? If they are MOS1, they are primed to fight. You give them the standard immunochemotherapy. If they are MOS2, they are detoxing your drugs. You need to add an AKR inhibitor to make the chemo actually work.
And if they are MOS3, they are hiding in the bunker. Standard immuno fails. You need to target the biology directly with FGFR inhibitors or by blocking that macrophage lipid pathway.
This study really feels like a huge turning point. We went from knowing that half of patients fail to knowing exactly which half fail and exactly why they fail at a molecular level.
It is a massive leap in mechanistic understanding. We aren't just looking at vague survival statistics anymore. We are tracking cellular behavior. We know that this specific gastric cancer resists therapy because it uses a metabolic disguise and leverages the immune system's own hunger for lipids against it.
It's terrifying, honestly, but also deeply hopeful because now we finally have targets.
Exactly. The knowledge of the pathway is always the first step to dismantling it.
You know, I want to leave you listening with a thought that struck me while we were reading about those APOA1 cells. It's something of a philosophical puzzle about cancer.
Oh, go on.
Well, the cancer cells in the worst case scenario, the truly resistant ones in the bunker, they are surviving entirely because they are mimicking healthy intestinal cells. They look like the good guys. They act like the good guys, processing dietary fats just like your body is supposed to do.
Right. Evolutionarily speaking, it's brilliant camouflage. The tumor is saying, I'm not a threat. I'm just part of the digestive tract.
It is brilliant, but it raises a massive, almost existential challenge for the future of medicine. If the cancer evolves to look and act exactly like the healthy organ it invades just to escape detection, how do we eventually kill the cancer without destroying the host's ability to process nutrients?
That really is the ultimate challenge of oncology going forward, distinguishing the flawless mimic from the vital original.
Right. It makes me wonder, Is the true future of cancer therapy actually found in metabolic disruption, like starving them or changing their diet, rather than just trying to boost the immune system? Because if they are relying on eating lipids to maintain their disguise and suppress the immune response, maybe we just cut off their specific food supply.
It's a very provocative idea. If the immune system can't tell the difference between friend and foe, maybe manipulating the local metabolism is the only tiebreaker we have left.
Something for all of you to mull over.
Oh.
Thank you for joining us on this deep dive into the microscopic battlefield of gastric cancer.
Always a pleasure to unpack this stuff.
See you next time.